Hi friends, my name is Nandita. I am from Andhra Pradesh.
Good morning, dear students. Today we are going to discuss about reproduction in organisms from chapter 7 CBSE syllabus class 10. Children, let us discuss about seed germination. Seed germination is the process by which a seed develops into a new plant. It involves the reactivation of metabolic pathways that lead to grow and the emergence of the radicle means seed root and shoot. Children, let us discuss the conditions necessary for germination. First, seed availability. The seed must be alive and capable of germinating. Second one is overcoming dormancy. Some seeds have dormancy mechanism that prevent germination until specific conditions are met. This may include exposed to certain temperatures, light conditions, or physical process like sacrification. Next, environmental conditions. Children, proper environmental factors are crucial for germination. Let us see what are those. First one is moisture. Water is essential to rehydrate the seed and active metabolic process. Second one is temperature. Each species has an optimal temperature range for germination. Third one is oxygen. Adequate oxygen levels are necessary for energy production during germination. Fourth one is light. Some seeds require light to germinate while other seeds best in darkness. Children, let us discuss the phases of germination. There are 4 phases in seed germination. First one is imbibition, second one is lag phase, third one is radical emergency, fourth one is pumule. Children, let us discuss in detail about each and every phase of seed germination. First one is imbibition. The seeds absorb water causing it to swell and swollen the seed coat is called imbibition. Let us see what is imbibition. Imbibition is the physical and physiological process by which seeds absorb water through their dry tissues primarily via hydrophilic colloids such as cellulose and proteins. This water's uptake is driven by a water potential gradient between the dry seed and its moist environment, resulting in a significant increase in seed volume and internal pressure known as imbibitional pressure. Children, now let us discuss about role in germination. The process of imbibition rehydrates the seed's tissue, reactivating metabolic pathways that were dormant in the dry seed. This reactivation leads to the synthesis of enzymes necessary for metabolizing stored food reserves, providing energy for the developing embryo. Additionally, the swelling caused by imbibition helps rupture the seed coat, allowing the embryonic root radicle to emerge and anchor the seedling. Children, let us discuss the factors affecting the imbibition. Several factors influencing the efficiency and success of imbibition. Seed coat permeability. The seeds with hard or impermeable seed coats may require sacrification, means mechanical or chemical treatment to facilitate water absorption. Second one, water availability. Adequate moisture in the environment is essential for imbibition to occur. Third one is temperature. Optimal temperatures vary among species, but generally warmer conditions accelerate the imbibition process. Fourth one is seed availability. Only available seeds with inactive embryonic tissues can undergo successful imbibition and subsequent germination. Children, now let us see the importance of imbibition. Imbibition is vital for initiating the germination process. Without it, seeds remain dormant and cannot develop into seedling. Understanding imbibition is crucial in agricultural and horticulture as it informs practices like seed priming and sacrification to enhance germination rates. Children, now let us discuss about the second phase of seed germination, which is called lag phase. After imbibition, the seed undergoes a period of metabolic activation without visible growth. This is called lag phase. The lag phase, also known as activation phase, which is the second critical stage in the seed germination process. Following the initial water uptake during imbibition, this phase marks the reactivation of the seed's metabolic machinery, preparing the embryo for growth and development. Children, let us discuss what happens during this lag phase. During the lag phase, the seeds undergo several physiological and biochemical changes. Let us see what are there. First one is metabolic reactivation. The seed's internal physiology is activated, initiating respiration and the synthesis of proteins essential for growth. Second one is enzyme activation. The hydrolytic enzymes are produced to break down stored food reserves such as starches and proteins into simpler molecules like sugar and amino acids. These molecules provide the necessary energy and building blocks for the developing of embryo. Third one is cellular repair and organelle activation. The seeds repairs any cellular damage cured during dormancy and actives organelles like mitochondria, which are crucial for energy production. Third one is preparation for radical emergency. Although there is a minimal water uptake during this phase, the seed prepares for the subsequent growth phase by initiating cell elongation and division process that will lead to radical emergency. Children, let us discuss the importance of the lag phase. The lag phase is vital for ensuring that the seed is metabolically prepared for the physical growth that allows. Without the successful completion of this phase, the seed would lack the necessary energy and structural components to support radical emergency and seedling development. Children, now let us discuss the third phase, which is called radical emergence. The radical emerges first, anchoring the plant and initiating nutrient and water uptake. Children, let us see what is radical emergence. The radical emergence is the embryonic root of the plant and the first structure to emerge from the seed during germination. Its emergence signifies that completion of germination and the beginning of seedling establishment. The radicle typically grows downward into the soil, anchoring the plant and initiating the absorption of water and nutrients essential for growth. Children, let us discuss the importance of radical emergence. First one is anchorage. The radicle develops into the primary root, providing stability to the seedling into the soil. Second one is nutrient and water uptake. As the primary root, the radicle begins absorbing water and minerals from the soil, supporting the seedling metabolic activity. Third one is growth initiation. The emergence of the radicle triggers subsequent growth phases, including root development. Children, radical emergence as a measure of seed vigor. Let us see what is that. In agricultural practices, the timing and uniformity of radical emergence are used to assess seed vigor. The seeds that exhibit rapid and uniform radical emergence are considered more vigorous, leading to better crop establishment. Children, let us discuss the fourth stage of seed germination, which is called the pumule. The pumule is the embryonic shoot present within a seed, consisting the epicoty, the stem above the cotyledons, and the first true leaves appear on it. Upon germination, the pumule grows upwards, emerging from the seed and developing into the aerial parts of the plant. Children, let us discuss the process of shoot emergence. After the radical, embryonic root anchors the seedling and begins nutrient absorption. The pumule initiates upward growth. This growth is driven by cell elongation and division into epicotyl region, pushing the shoot tip towards the soil surface. The pumule exhibits negative geotropism, growing against gravity, and positive phototropism, growing towards the light. As the shoot emerges, the cotyledons, means seed leaves, may either rise above the soil surface or remain below, depending on the plant species. Children, now let us discuss the role of light and photosynthesis. Upon reaching the light, the pumulus leaves expand and begin photosynthesis, producing energy for further growth. This transition from relying on stored seed nutrients to photosynthetic energy marks a critical step in seedling development. Children, now let us discuss the final stage of seed germination, which is called seedling establishment. Seedling establishment is the concluding that critical phases of seed germination where the young plant transitioning from relying on seed reserves to becoming an independent, means photosynthetically active organisms. This stage ensures that seedling survival and sets the foundation of healthy plant development. Children, here are some key process during seedling establishment. First one is root system development. The primary root radicle extends and branches out, forming a network that anchors the plant and absorbs water and nutrients from the soil. Second one is shoot growth. The shoot pumulae contains to grow upward, developing stems and leaves that will carry out photosynthesis. Third one is photosynthesis initiation. As leaves expand and mature, they begin photosynthesis, allowing the plant to produce its own food and energy. Fourth one is transition from seed reserves. The seedling gradually shifts from utilizing stored nutrients in the seed to relying on photosynthetically derived energy. Children, as we discussed earlier, let us see once again what factors influencing successful seedling establishment. First one is soil conditions. Adequate moisture, proper aeration and nutrient-rich soil support root and shoot development. Second one is light availability. Sufficient light is essential for photosynthesis and healthy growth. Third one is temperature. Optimal temperature promote enzymatic activities and metabolic process necessary for growth. Fourth one is protection from stressors. Minimizing exposure to pest, diseases and environmental stresses, enhancing survival rates. Children, now let us see the importance for seedling establishment. The successful seedling establishment is crucial for plant survival, which ensures that young plant can sustain itself and continue growing. Second one is crop yield. In agricultural, uniform and robust seedling establishment leads to higher productivity. Third one is ecosystem stability. In nature settings, healthy seedling establishment contributes to plant population maintenance and biodiversity. Children, finally we discuss that even factors which are influencing for germination. First, the seed treatment. Some seeds benefit from treatment like soaking to soften that seed coat, especially those which thick outer cell. Soaking can help the dormant embryo break through more easily. Storage conditions. Seed storage under optimal conditions means cool, dry and dark, maintain higher availability. Age and improper storage can reduce germination rates. Understanding the germination process and factors that influence it is essential for successful seed propagation, whether for agricultural, horticultural or ecological restoration. Thank you.